Hi, Rachel. Thanks so much for joining me today. I'm really excited to talk about your research.
Yeah, thanks for having me. I'm also excited.
So, to start, what sparked your interest in neuroendocrine tumor research, and how did your early work in plant pathology and process chemistry really shape your approach to translational medicine?
Yeah, so my journey with science and what is now my career in studying neuroendocrine cancer started, well, probably in childhood when I loved math and science and figuring out how everything in the world around me worked. And that really gained momentum in college when I got started in research in plant pathology. We studied Phytophthora infestans. It caused the Irish potato famine. It infects potatoes and tomatoes. So I got to study the genetics of this disease, and that's where I learned a lot of the basic laboratory techniques, how disease pathology, disease genetics work. And even we studied how that disease spread across the globe in the 1800s by studying archaic DNA. So, right there, I was already like, this is awesome. This is so cool. I had found something that I loved. I was in the lab late at night just to finish an experiment because I loved what I was doing. And then on top of that, I also applied some of my knowledge that I was learning in chemical engineering to study process control and water purification. And that experience also taught me a lot of hands on techniques and applications. It built my confidence in what I could do. And it was those two experiences that I really enjoyed. And it made me realize I wanted even more of a challenge. Now, don't get me wrong, both of those fields are very challenging. But I wanted to take what I had learned and apply it to something that I felt passionate about, which was cancer research. And so that's when I decided to go and get my doctorate in cancer biology at the University of Alabama at Birmingham. And I'd like to believe that everything happens for a reason. Some things I can explain, some things I can't. And I happened to just end up getting connected with a lab that studied neuroendocrine cancer. And at the time, I had never heard of neuroendocrine cancer. But the more I learned about it, the more fascinated I became. I realized that this field uh, needed and still needs research. It was the ultimate challenge for me to want to solve. And turns out I'm still trying to figure out this disease. Gosh, like 8 years later, and it was those early experiences that got me interested, made me fall in love with science even more to take on the ultimate challenge of trying to study neuroendocrine cancer.
And that's one thing that's come up a few times in the conversations we've had on this podcast where you really need to be able to pull expertise and gain skills in a lot of different functional areas. Like to be a great chemist, for example, you have to be able to utilize math and physics, and you really need to build your skills in a lot of different areas. So that makes a whole lot of sense to me in terms of hearing about where you came from and what was interesting to you, and then applying it to this area that you focused on for 8 years, I believe you said.
Yeah, it's been 8 years. And I still go back, I'm like, oh, I did this, back in college, and it still comes up. However, I used to do a lot of differential equations and derivatives in college, and I don't think I could do that now. There's definitely skills that I have lost, but I have gained many more.
Totally. I'm sure we can all relate to that, especially now in the age of AI, right? Doing things differently, and you're going to lose some of that. Oh, I used to do this by hand in Excel type.
My gosh, I'm even seeing that now in terms of summarizing information or even a literature review. If I want to know, everything that's been done about this really niche topic, back in my day, I had to look it up. One Google search at a time, which I know is different than back in the day when they had to go to the library and find a book. But now people can just ask AI.
Yeah, it is wild for sure. And now I know for my next question that some of this is in process, so I hope I don't butcher the right terminology. But how would you describe just the general process of moving scientific discoveries from that basic research to more of that clinical trial or phase 1B investigator initiated trials, which is a totally new topic for me, if you could get into that?
Yeah, absolutely. This is where I get really excited about my research is the fact that we can do studies in the lab, very basic science type level stuff, and then drive that all the way to something like a phase 1B trial or something with a human endpoint. And my experience with that has been made possible because of my collaborations with particularly surgeons and other physicians. I'm the PhD, I'm the lab nerd, I'm in the lab all day, but I work with some really brilliant clinicians, physicians, surgeons who are out there interacting with patients, and their investment in the research is what helps drive it from the classic saying bench side to bedside um, type situations. My conversations with them, my collaborations with them are fascinating, and it's what keeps the work, in my opinion, very relevant to the patients, because I of course have a million questions I could ask, and they help direct my energy and the questions I ask into what's most applicable to patients. They know what needs to be done. They know the biggest needs of the patients because they're seeing these people every day. And so then they can guide me like, hey, Rachel, the patient really needs a drug that stops metastasis from continuing to spread, for example. And so I can direct the efforts in the lab to the greatest needs of the patients. They're also very helpful in terms of recruiting patients, explaining research to patients and why it's important, getting patients involved in studies, which a lot of patients are very excited about, and I'm very grateful for that. Many patients are quick to help and they want to be involved. Just donating a piece of the tumor to research, that can go a long way in the lab if they consent to donating just a piece of their tumor that would be taken out by a surgeon. I can go in the lab and then dissect that. My collaborations with physicians, surgeons, is what makes my work very translational, which I like to see. I was trained as an engineer, not technically as a biologist. I like to see it to believe it. With this translational research, we go from an idea in the lab, and it takes a lot of time and a lot of work and a lot of safety testing to go from something in the lab to something that's going to actually interface with a patient.
That makes sense.
So I feel like I talk about it kind of casually, but it is also a lot of hard work and time and a lot of people involved and regulatory documentation and all of that.
Somewhat straightforward being able to connect your work to this podcast because you do utilize mass spectrometry. How has the integration of mass spec impacted your workflow in your cancer studies? And what are some of the most critical analytical challenges you face when profiling these tumors?
Yeah, those are great questions. So I've utilized mass spec from very basic science mechanism stuff to using it to actually study human tumors grown into mice. So we have used it on the basic science side of things to answer questions like what specific transcription factor is binding this sequence of DNA in the promoter region of Notch3 to drive this tumor suppressor in neuroendocrine cancer. I have answered that question with mass spec, which is very niche, but also very important. But At the same time, we use mass spec we put human neuroendocrine cancer cells into a mouse that didn't have an immune system, of course, grew those tumors, and actually had a couple groups of tumors. Some that were just wild type, and then some I had used CRISPR to engineer some of the tumor cells and then put these different groups into mice and then saw how they grew differently or how they behaved differently. And part of the analysis was using mass spec to identify proteins that were differentially expressed between these different groups of neuroendocrine tumor cells that I had engineered with CRISPR. And so we were able to find differential protein expression. Actually, it'll be published soon, that I can hopefully soon share with everyone our exciting findings on what was different between those two groups. And so mass spec has been an excellent tool for me in my studies, and I collaborate with Dr. Mobley here at UAB. He's awesome, and he's a leading expert in mass spec and has helped me design these experiments. And so having someone who is an expert has made my studies successful, because as much as I'd like to know everything in the world, I can't. So if I can work with someone who knows their area of expertise and I have mine, we work together to design these experiments so that we can get these answers. I'm no expert in mass spec, but he was teaching me all about the ionization and fancy stuff, and the equipment looks really cool. I got a tour of that. So that's been really exciting and a valuable tool for us. But I will say, studying an uncommon cancer like neuroendocrine cancer is difficult. I do an experiment using mass spec, studying a neuroendocrine tumor, and I get a result and I have no idea what it means. I can't say, oh, I wonder what this person did, or I wonder what someone had done in the past in this scenario. I can't ask those questions because it's never been done before. So I don't have a lot to go off of. Now I can try and look at other cancers and extrapolate and apply problem solving from other cancers to neuroendocrine cancers, but it's rarely that easy. And what I've learned is neuroendocrine cancer often does not behave like other cancers. And so a lot of the work that we do is pioneering. And so that takes a lot of communication and honesty, and one step at a time. There's not been a ton of work done using mass spec in neuroendocrine tumors. I mean, there's definitely not enough research at all in neuroendocrine tumors. This is very new. But that also makes it very exciting, because when we do discover something, it's new and it's contributing to the body of knowledge for this cancer type.
This might be hard to answer, but are there any data insights that were only made possible because of advanced mass spec methods?
Definitely. I think answering some of those very basic science questions, like what transcription factor is binding this DNA region to drive gene expression of this gene that's controlling the growth of neuroendocrine cancer, we need mass spec to answer those questions. And I won't be too controversial, but using mass spec versus maybe RNA sequencing has a lot of exciting applications. They're both very good techniques. There's a lot that happens between RNA and protein. So using mass spec to profile and to understand protein expression in a tumor is very valuable. I won't say it's more valuable than RNA sequencing, but I think it's something a lot of people should be doing, and it could help facilitate very important discoveries by understanding protein level dynamics in a tumor. And those type of questions can be answered with mass spec.
One comment you made when we were getting ready for the interview was that you thought that spatial mass spec was really cool and a technique that in the future you hope to employ one day. I was just curious why it stood out to you.
Oh yeah, that's so cool. I hope one day I get to use that. So tumor heterogeneity is a big deal. I mean, it's been a big deal for years, but we know that these tumors, any tumor, not just neuroendocrine cancer, are very complex, very heterogeneous. And inside of a tumor, it's not just tumor cells. You've got blood vessels, you've got endothelial cells, you have immune cells, you have different type of immune cells, you have dividing tumor cells, you have dying tumor cells, you have all sorts of stuff going on in that tumor. And so using something like spatial mass spec, where you can look at the interactions and the relationships of these different cells, I think is going to be a game changer. It's not like you're just taking the tumor out, mashing it up, homogenizing it, and then analyzing what comes out. If we can preserve the structure and the architecture of the tumor and all of the heterogeneity and all the different cell types, and then use something like spatial mass spec to analyze those intact relationships, I know that's a game changer, and I think that's so cool, and I hope one day that would be something I could do in the future.
That's very cool.
Yeah. So, like, for example, pancreatic neuroendocrine tumors do not have a lot of immune involvement. Immunotherapy has been a great breakthrough and proved to be very effective with other cancer types like melanoma, but it's not proved as promising in pancreatic neuroendocrine tumors. And these tumors, they just don't have a lot of immune system involvement. And obviously, the question is why, and I think that's still being answered. But if we could use something like spatial mass spec to preserve the immune tumor cell interaction, that spatial relationship, and better understand what's going on between the interface of the immune system and the neuroendocrine tumor, that will be very exciting and could open up many avenues of research.
Earlier, you touched on the general overall process, but what advice would you give to scientists or clinicians who are aiming to bridge that gap between laboratory research and the practical clinical application, especially in rare cancers?
Yeah, the best advice is to talk. And that's hard sometimes as scientists and even as clinicians. We tend to be introverts, but having those conversations, building those relationships, maintaining those relationships, because when we all come together, we often have the same goals and we want the same things, but we can't do it all by ourselves. We have to rely on each other to reach our shared goals. With the groups that I help lead, the Net Models Consortium and the Net Cancer Foundation, some shared goals between these groups are bringing together scientists, clinicians, physicians, and patients, putting all these people in the same room and fostering those connections and those relationships to work together, because that's how we're going to make progress. I need the surgeons to be able to get tumors physically out of the patients. I need the patients, most importantly, and I need them to understand and be invested and contribute to the research that we're doing and help drive those directions of those research. And then we need the scientists in the lab to do the analysis. So everyone has their own expertise and contributes to the overall goal. But we have to meet these people. And that's really step one, is finding the right people, meeting them, getting connected with them, and then staying connected with them. I will say it's not always as easy. There's always administrative barriers, life barriers, especially when we start talking about international collaborations. That adds a whole other layer of complexity that's real. And so we kind of have to be prepared for there's challenges. There's going to be ups and downs. There's going to be paperwork and all that type of stuff. But not giving up, and then I think, the fact that most people have shared goals and realizing how working together reaches those shared goals is what keeps driving a lot of these collaborations. My advice would be don't try and do it all by yourself.
No, that's great advice. And before we started recording, you had touched on your travels as of late, but you just provided a great segue in terms of how do you balance advancing your research, engaging in public advocacy, and then you just mentioned the consortium you're involved with. It's a lot to try and do all at once. So, any advice there for people?
Oh yeah, no, it's a lot. It's been highlighted this month because I've been traveling so much of just how much I've got going on. But all of it's something that I'm passionate about and that I enjoy doing. So that definitely helps. This month, I was so fortunate to travel to Oxford, England for the 2025 Net Models meeting that was sponsored by the Net Models Foundation. Both are nonprofits that I'm involved in. That meeting was incredible. We were able to bring together neuroendocrine tumor research experts and more than that, we were able to bring in vets, PhD scientists, clinicians, patients, everybody in the same room and talk about some of the biggest challenges in the field and what are we going to do about it. So that was an incredible experience. And so that got me excited and, I'm like, this is why I'm doing this. And so that kept the momentum to keep going. And then fortunately, back in the lab, when I do travel, I don't stress too much because I work with an awesome surgeon who helps me run the lab. And then I have awesome people in the lab that are helping get the research done. And so I can go to a different continent and know that the team that I work with is going to keep the research going forward. Having a good team is awesome. I'm passionate about all that I do. I think the research that we do in the lab is really important, but also at the same time, the patient advocacy and the consortium and the international efforts to bring people together is going to pave the future for the research that we're doing in the lab, which is really exciting. But also, like I had mentioned, the cost of all of this is maybe some lack of sleep. I haven't folded laundry in maybe 2 weeks. But having the community and all of my friends and colleagues support me makes it all happen. I'm very grateful for all that.
You and I have very different job roles, but I relate to a lot of what you're saying, whether it's coming back from a conference and having that excitement about something new that you've learned, or just the need to be out there in the market, which for me is visiting customers and laboratories who are actually using organomations, evaporators, because all of those experiences are really eye opening and lead to a really useful cycle, I suppose, in terms of inspiration and being able to push your work forward in general.
Yeah, it's necessary. And I've seen over the years, and especially during my PhD and my postdoc, you're spending weeks in the lab as most experiments don't work. You got to pick yourself up, do it again. And this goes on and on. And you can start to get burnt out and ask questions like, why am I doing this? Does this even matter? And then you go to a conference, you go to a meeting, you're surrounded by people that are understanding what you're going through, and you have conversations, and you're like, oh, this is why I'm doing this, or like it is possible, and I do have support, and I'm not alone, and it's gonna be okay. And then you come back to the lab, and then sometimes it can be a cycle of like inspiration and then despair. And I'm sure you have your fair share of experiences with that.
So, what did we miss that you would like people to know about your work? Any topics we didn't cover?
We covered a bunch. I think one of the most important things I can convey is that neuroendocrine cancer is a thing. Most people probably have never heard of it. It's not brain cancer. It can show up all over the body in various organs. The symbol of neuroendocrine cancer is a zebra, so you'll probably see a zebra here and there. So like breast cancer is pink, and it is Breast Cancer Awareness Month. It's October. Neuroendocrine Cancer Day is November 10th every year, so that's coming up. There's zebras because there's a adage in medical school. Now I've heard about this, but in medical school, they teach you that when you hear hoofbeats, think horses. And I hope I don't butcher this, but my best understanding of this is for people to think the most common solution. So instead of going down some rabbit hole about this, rare disease that someone might have, if they present to you with these symptoms, the answer is probably the obvious one. But that's not the case with neuroendocrine cancer. Neuroendocrine cancer presents with symptoms that are very common. It can take 5 to 7 years for a patient to get diagnosed. Sometimes patients get diagnosed with anxiety before they get diagnosed with neuroendocrine cancer, but the symptoms are very vague and can be attributed to other things. And then someone's probably not going to think, oh, you have this uncommon tumor. So in the field of neuroendocrine cancer, it's when you hear hoofbeats, think zebras, think something unique. I like to bring awareness to neuroendocrine cancer. We definitely need more funding, more science. It's a fascinating disease that there are a lot of dedicated people working to understand it. And every discovery is an important discovery.
I totally agree.
And mass spec can help us make important discoveries.
There you get a profile of the tumors for sure.
Exactly.
Well, Rachel, thanks so much for the time today. And maybe once everything is further along, as you noted, maybe we can have a follow-up conversation on how things are going.
Yeah, I would love that. Once I'm going to get, hopefully, I'm saying this out loud, I'm going to get this paper published. And we can talk maybe about some of the exciting discoveries, especially, what we found with the mass spec.
That would be great. I'm looking forward to it. Thanks so much.
Of course. Thank you.